So in this one, we're going to discuss the fluid mosaic model. And so this is the idea. So recall that we talked in the last video about how materials, lipids, and things incorporated into the lipids can move laterally. So this is a video showing the lipid bilayer. So these are, here's the polar head groups, and then the green are the tails, and they're pointing towards each other to form the hydrophobic layer, and then the polar head groups are sticking out. So in this video, you have the lipid bilayer, and then what you see here are oligosaccharides in purple that are part of the membrane surface. Coming in, showing in green, are what are called integral membrane proteins, which span the lipid membrane and can facilitate things such as diffusion or transport of different molecules across the lipid membrane. And so we'll discuss that more in detail in the next chapter. But the idea is that there are different things that incorporate, and for the point of the fluid mosaic model, what's important is that, again, proteins and other components of the membrane can move laterally through the membrane fairly easily. Whoops. Let's end that. And so let's look at a static picture of the plasma membrane. Okay, so what might these components then be that can move through the membrane? And so again, in this figure, the blue spheres are going to be the polar head groups of the phospholipids that make up the lipid membrane. The tails are shown sort of in these wriggly lines, so expect something that looks like this. All right, each of that is a phospholipid. The purple are different types of proteins. Some of these might span the lipid membrane, so this is the membrane. Okay, some of these proteins, might span the entire lipid membrane. Okay, these are called integral membrane proteins. Okay, and then there are other proteins that might just be associated to the membrane, or maybe even to other membrane proteins. Let's make that a different color. That's more clear. All right, these are called peripheral membrane proteins. And then another type of proteins that you may find are proteins that are covalently linked to a phospholipid, for instance. So we have a covalent linkage. All right, this is a lipid linked protein. Okay. So this gets at the idea again that remember, proteins and enzymes do a lot of the work in biology. They do a lot of work in the cell, and so there have to be different proteins that are incorporated into the lipid membrane in order to facilitate things, again, like transfer across the membrane, or different processes that are important at the surface of the at the surface of the cell. Excuse me. And so on either side of the lipid membrane, you have an extracellular side, so that's outside of the cell, and then cytoplasm, which is inside the cell. The green sort of chains of geometric shapes are meant to represent different oligosaccharides, so glycolipids, and oligosaccharides, which are chains of sugars, okay, like we learned in our recent work on the carbohydrate chapter, where you can have different oligosaccharides. And what these act as are sort of as barcodes, which can be used in order to differentiate one type of cell from another. So, for instance, this is important for immunity, so the immune response for being able to detect different types of cells, or even viral recognition of cells that the virus are meant to specifically interact with, for instance, okay? And so there are a lot of different components, the point being there are a lot of different components of the membrane, and together we call this the flow and mosaic model, okay? And they can move laterally. Now, in addition to that, in addition to this asymmetry within the lipid membrane, so a lot of things are incorporated in the membrane, there's also asymmetric phospholipid distribution. So remember, there's an inner leaflet and an outer leaflet. So again, if we call this the extracellular space, and then this is the cytoplasm, we can also think of, because there's a bilayer, there's an inner leaflet and an outer leaflet of the bilayer, okay? And different phospholipids specifically integrate into these inner and outer leaflets. So this chart shows, for instance, several different types of the lipids that we talked about in the earlier videos, such as sphingomyelin, phosphatidylcholine, phosphatidylethanolamine, and phosphatidylserine. And so if you look at this, what you see is that at this chart, you see that sphingomyelin, phosphatidylcholine, there are way more of these in the outer leaflet than there are in the inner leaflet. So there's a higher percentage on the outside of this leaflet. So there can be some asymmetry in this. And likewise, phosphatidylethanolamine and phosphatidylserine are more dominant on the inner leaflet, okay? And that helps facilitate different functions that are necessary for the outer leaflet and the inner leaflet. And again, that's important because the outer leaflet interacts with the extracellular space, whereas the inner leaflet interacts with inside of the cell. Integral membrane proteins, which again we briefly discussed, so are tightly membrane associated. So they span, for instance, they can span the membrane, and so remember that the inside of the membrane, inside the lipid bilayer, is hydrophobic, whereas the outside is polar, which means that membrane-associated proteins are also amphiphilic, just like phospholipids, where the ends here are going to be more polar or charged residues, whereas inside they'll be nonpolar. Okay. And so you can see that in this figure on the right-hand side, where acidic, for instance, acidic or basic residues, amino acid side chain residues, are shown by either blue or red. And you'll see that on the outside, well, let's back up. So the green are, for instance, phospholipids that interact with the core of the integral membrane protein. On the outside are the things that will stick, or the sides or the faces that will stick out towards the extracellular or the cytoplasmic face. And then what you see is that there are more, by looking at the red and blue incorporated, there are more basic and acidic residues at the facing out towards the outside, towards where you would expect to see the sides that face extracellular or the cytoplasmic side. Versus the inside, there are very few blue or red patches. In fact, there's just gray, which indicates some sort of nonpolar residue, such as isoleucine, leucine, or tryptophan, or residues like that. And this is in stark contrast to, let's say, a soluble protein. So this is hemoglobin, shown here on the right, which we've studied quite a bit. And that, of course, is a soluble protein that's not integrated into a membrane. And what you see is that there's an even distribution of red and blue patches all over the protein. And that's consistent with this needing to be, for instance, in the cytoplasm and needing to be soluble and not within the membrane. Okay. And so, again, consistent with that, this is another way of looking at that. So if you're looking at the polarity of an integral membrane protein, again, this is going to be amphiphilic. And so chains that reside either in the cytoplasmic or the extracellular. So in this diagram, the left-hand side is outside the cell, so that's extracellular, versus the right-hand side will be the cytoplasmic side. And so there's more, you see more, for instance, aspartates, glutamates, histidines, arginines, lysines in the chains that are sticking out into these soluble parts. Whereas the region that goes through the membrane, so known as a transmembrane domain, has more hydrophobic residues such as leucine, isoleucine, glycine, alanine, and there are very few instances of charged residues or polar residues that are seen in the chains that actually go through the transmembrane through the membrane. And so some domains, some different secondary structures that may be seen that are important for allowing for these transmembrane domains, for instance, are alpha helices. This is a common secondary structure seen for transmembrane domains. So this is shown for bacteriorhodopsin, which is an integral membrane protein, and this is a bundle of alpha helices, so 7 alpha helices, which span the transmembrane, and they wind through the membrane. So if we start from the blue, it's, for instance, we'll go through the membrane. Part of it will bend back in. And I'll do this 1, 2, 3, 4, 5, 6, 7 times. So there's 7 different transmembrane domains, and then there are soluble loops on the outside, on either side of the membrane. Okay. Another motif that you might see are known as Betabarrels. This is Betabarrel for the protein OMPF, and so this, in other words, is not restricted to one type of structure for proteins to come. For proteins to be integrated into membranes. There can also be hydrophobic secondary structures which which use beta sheets instead of alpha helices in order to create the hydrophobic core which is necessary for an integral membrane protein. And so this allows for creation of a protein like this, which is important for transfer of, which is actually like a pore, which allows for transfer of materials in and out of the cell. So the point on all this is that there are a lot of different components of the lipid membrane. Some of these, for instance, are proteins. We can also think of course, the phospholipids and the different types of lipids that will integrate into there. There's also lipids such as cholesterol, which integrate, and that can have different functions, for instance, on the fluidity of the membrane, and also what can be incorporated into the membrane, including the proteins which interact directly with these lipids. And so the stability of these proteins are very important on the composition of the lipids that are in there. And so these all act together in order to allow a different functionality for these proteins and for the lipid membrane.